good. How was yours? So, can we just start now?
Yeah, go ahead.
Okay. So, can you explain to me why we're here today? What's the gist of your research? Is it as you would explain it to a 12-year-old?
12-year-old. So, what are we doing here is we analyze tissue specimens taken from sick people, and we use a new device which is called gene chips. And we developed a system that by reading changes in the genes of the sick tissue to understand the disease processes better and earlier. And the current standard was to use the microscope, which was invented 150 years ago. And this is essentially the next generation beyond the microscope, because you can't see the molecules in the microscope. And with this device, we can read the molecules in the microscope and analyze the results in sick tissues together what we see in the microscope, and by this improve the understanding of disease processes.
So you can see what's making why tissues are sick.
Why and how sick they are, and how good they will respond to what therapy, and what your prognosis is. And this with a much broader level of information. And the challenge was you get more than 50,000 points of information from this little device now, and we can run hundreds of specimens in a month here. And the challenge was to understand this large amount of information, and that's why we had to develop software algorithms to interpret this in the right way, because a lot of changes in the genes are like fever. So we all have some fever, but not all of us with fever have a very sick situation or are going to die. So to get it into the right context, how sick you are, what your disease is, how you will recover, whether you will recover at all, that was the challenge. Not to run this device, which is a commercial product, but to understand the large amount of data you get and interpret it in the right way for the clinical partners, and treating them, the patient, in the right way.
So these algorithms or this computer system helps you to understand all the data that comes pouring in.
Yeah, and we essentially summarize this into 8 or 10 numbers and can visualize that. And this gives the everyday clinician on the ward the possibility to use this new technology in patient care. And we plan, or we will start now, a multi-center international study using this in the real clinical setting, with centers in the United States and in Germany, the UK and Spain, and give clinicians reports of all these 50,000 data points. They can use it day to day. You can imagine if you get a spreadsheet with 50,000 data points, it's useless to somebody who has really to act on the patient. And so he gets a set of numbers back and a graph where he can see where this patient's molecular changes are falling, and he can interpret it like a fever measurement or a specific answer, and then draw his treatment conclusions.
And so how will that ultimately benefit patients on a day-to-day basis? Is it better health care, or what? How does it help patients?
Overall, it is better health care, and in particular it is more precise diagnosis, further improved diagnosis. It gives you a stage, so how you have a certain disease, but how sick you are with this disease, how advanced. And that then triggers a more specific treatment. So all treatments have side effects, but when you are very sick, that justifies to give you a more intense treatment. So these decisions are improved, and by that, outcome will be improved as well. More specific and more appropriate treatment.
How many microscope tests would you have to do to pull out the same equivalent amount of information off a gene chip?
You will never ever come with a microscope to that level of information. It's impossible. This is the imagine what we see with our eyes now is the first level of information. So when you're red in your face, I know you have fever. Then I take a specimen from your enlarged lymph node, because I think the disease is, and then I look at the cells. That's what the microscope is doing, the next level of information. And this device then takes the cells from that tissue and looks at all molecules changing in that. It's the level below. It gives you another level of information which is not accessible by the microscope or the human eye otherwise.
It takes it to a higher level, it goes deeper.
Yeah, and essentially the resolution of your eye now is probably 1 to 4, with a microscope is 1 to 1,000, and this goes down 1 to 100,000 on a molecule level.
And is anybody else in the world using algorithms?
Yes, there are in other diseases, using it, for example, in cancer. There are approaches and studies going on, but we are the only group essentially in the world using it in organ transplantation and inflammatory diseases. We now start also a project in inflammatory bowel disease, where we will support the histological diagnosis.
And how important is this to use it for organ transplant patients, because there's so many things that could go wrong with them after a transplant?
Yeah, so today the surgical part of an organ transplant is essentially standard and has 100% success. And the challenge is in that you want to live with the transplanted organ for the rest of your life. But over 10 to 15 years, half of the organs fail, and we do not understand really why. And with this device, we already showed that we can assess diseases operating in transplants, specific for transplanted organs, can be diagnosed better and earlier, and we can assess prognosis much better and therefore guide therapy in transplant recipients more appropriate.
And how...
Pressure's on.
And what's the correct spelling of your name?
Like the correct spelling of my name?
Yes.
M-E-N-G-E-L.
And Michael is normal.
Michael is normal. Normal Michael, yes.
And what's your title?
It's my title. I'm a researcher at the University of Alberta, and I'm also with Alberta Health Service as the section head of transplantation pathology.
And how many patients do you hope will be involved in this international study?
500.
500, and how many years will it take?
The trial would take just 1.5 years. To recruit, probably around a year, 1.5 years, because these are essentially the largest transplant centers in the world are participating in Germany and the UK and in Spain. And then the data analysis probably takes another year, and we also have for this trial an industrial partner and who is very much interested to implement this into the clinic as fast as possible.
So do you think we could see physicians in organ transplant and inflammation specialties using this in the clinic every day in a few years from now?
Yes, and we expect it in the next 5 years to have this implemented.
And medically speaking, how fast is that?
The turnaround time when we get a specimen is currently in this lab setting 2 days to get the result, which is reasonable. Histology is still faster, but with the economy of scale, you can assume in the further development of the platforms that this will happen much faster, within essentially hours to get the result.
And typically from a research lab to go to the bedside, it can take, isn't it usually 25 years? So 5 years is quite quick, isn't it?
Yeah, no, this device was invented probably 15 years ago now, but it is now that standardized and commercialized that we essentially we order it, it comes. And the last step was to implement that in a way that an average clinician who is not a basic researcher can use it for his patients. And that was the last step to get it from the bench to the bedside. And the whole project to understand what we get from this started in 2004 with the Genome Canada grant. So it was 6 to 8 years to make it really then from the first clinical research to patient application.
And you talk about sort of give 8 bars of information, or 8, sort of break it down to 8 areas of information. Can you give us an example of what some of those areas of information might be?
We summarized the molecular information, the changes in your genes, in your sick tissue, into the major biological processes which characterize diseases in transplant. And that is inflammation, and that is different types of inflammation. You have mediated that by certain cells which have different functions. And that is injury. All your tissues when you get sick have a certain injury response. And there is a set of molecules which you have normally. So all your cells have a normal function in the process. And when this is not working, you know it when you have a sore leg. You can use it very well. And the molecular changes behind that means that a lot of molecules go down. They are not functioning, and we can measure these and can tell you how much they go down, and that correlates with how sick you are. Yeah, so that gives you again an idea about your prognosis. So when you're very down with your genes, so you have a worse prognosis, and then we can give this. And the biggest advantage is this is a very reproducible and robust measurement. And when pathologists look at a microscope, there is a piece of subjectivity. Also, when a doctor looks at you and you look at your child and say, oh, you do not have real fever, it's a bit. So, but that's subjective. One mother is already concerned, and in the emergency room the other mother still sends it out for hockey. But this measurement is a number, and it's reproducible, and it can be standardized, and every patient can be plotted on a scale and say, look, in the last 500 patients you're out here. That's your risk, and that's your probability that you are that sick.
So measure the sickness of different molecules in the tissues.
Yeah, and it is very, very standardized and robust between centers.
And what kind of molecules do you look at? Like molecules responsible for what areas?
Yeah, as I said, for inflammation, for your T lymphocytes, for your macrophages, for your B lymphocytes, for your blood cells, like which are responsible for your microcirculation of blood and your oxygen support for the tissue. And then we look in the local cells, which are just specific for one organ, like in the kidney or in the heart, it's your heart muscle cell, and we can measure how much they are down in their energy transport, in their energy generation, and that correlates with your heart function and with your kidney function and your prognosis when we have identified the disease, whether you recover.
And is there anything else you'd like to add that I've forgotten?
No.
Excellent. Put anything else?
No, I don't think so.